Welcome to our Cloning and Primer Design in Lasergene Core Suite webinar. I'm Katie Maxfield, and I will be hosting the webinar today. And I'm here with Brian Anderson. And Brian is going to be doing the majority of the webinar today in a live demonstration of the Lasergene software. And to start, he'll give you a brief overview of cloning and primer design and Lasergene Core Suite. Before he gets into that, I just want to let you know that we do want you to ask questions along the way. Your phone line has been muted, so to ask questions, we ask that you chat them in to the chat dialog in the bottom right-hand side of your screen. Or if you don't see that, you may just need to open the chat dialog by clicking the chat icon on the toolbar in the top right. Any questions that we don't get to in the webinar, we'll be sure to follow up with individuals after the webinar to make sure we get all of your questions answered. So with that, I'm going to turn it over to Brian, and he'll tell you about cloning and primer design in Lasergene Core Suite.
Thank you, Katie. Hello, everyone. My name is Brian Anderson. I'm one of the technical sales scientists here at DNASTAR. I've been with DNASTAR for just under 7 years. Today, as Katie briefly mentioned, I will walk you through some of the logical steps to consider when performing in silico cloning and primer design in Lasergene. The format of today's webinar will be a very, very brief PowerPoint, followed by some projects within the SeqBuilder application. Okay, let's get started. All right, I'm going to show my PowerPoint here. A little bit about DNASTAR first. DNASTAR is based in Madison, Wisconsin, home of the University of Wisconsin-Madison. Our company basically was founded on the research, or it came out of the laboratory of Dr. Frederick Blattner at the University of Wisconsin-Madison in the early 80s. Dr. Blattner is most known for being the first researcher to sequence the E. coli genome at the time, which was a pretty significant undertaking. The core tools that what we today call Lasergene were instrumental in assisting Fred's team in sequencing and further characterizing that E. coli K12 genome. A number of these researchers that are seen there in that paper are actually still actively supporting and developing DNase products to this day. That firm foundation established by Fred's research, those tools, as well as acceptance as a novel tool by researchers in the literature, and our award-winning customer support have made Lasergene the most widely cited sequence analysis tool currently available today. This is basically just a brief graph that kind of shows the running total, the total number of citations out there in the literature. So as you can see, since the early 80s, we have over 8,000 citations in the literature today. As mentioned previously, customer support is one of the reasons why researchers tend to use our products. An example of this support would be built into a software tool that we've recently introduced to Lasergene called our DNASTAR Navigator. Basically, if you're looking at some of those screenshots, Navigator is a resource that will provide you quick links to the various tools that we do provide in Lasergene. There's also a help index on the right-hand side there that shows you direct links to all of our help menus that are built into the software, as well as the ability to do Boolean searches. So if you wanted to search within, say, SeqBuilder and search, say, cloning at the same time, it will search all the different applications as well that fit cloning as well as SeqBuilder, the software. Today we'll go over some of the following logical steps. Working with the target sequence, what things within the software can I do to make sure that my target is optimized for that specific expression that we're working with. Working with the vector sequence, looking at the multiple cloning site of that vector, making sure that the vector is in frame. Creating selectors to basically of enzymes of interest that are specific for my multiple cloning site. Looking at traditional cloning, as well as some TA cloning, as well as some of the other in silico cloning wizards that we have, as well as creating the suitable primers to insert those restriction enzymes toward the end of your target to make sure that those clones will be successful. All right. With that in mind, let's just get to the software. So I'm going to minimize my, and then everyone should be able to see my screenshot here. Before we actually get into the project, I thought we'd kind of show how you can launch the software, the Lasergene software, for those individuals out there who may not be that experienced with Lasergene. As previously mentioned, we have that what we call the DNASTAR Navigator. The Navigator can be basically available from the Start menu, or in this specific case, I've previously pinned it to my Task Manager here. So simply just clicking on it will open up that Navigator interface. As you can see here, in this Navigator, there's direct links to all the different applications that we do provide. So whether you're doing next-gen sequencing, we have a tool for next-gen sequencing. Microarray analysis or RNA-seq or ChIP-seq analysis, we have tools for those applications. Or in this specific case, we're going to be focusing on the Seq Builder module. We'll show that link directly to the Seq Builder application. One thing I just want to briefly mention here is if I simply highlight over any of these icons, there's direct links to additional resources, so videos online. So we have over 120 different videos on our website and our video archive. FAQs, tutorials, web tutorials, video tutorials, as well as PDF tutorials that you can download and work through, as well as demo data to get you a better understanding what the software can specifically do. So let's just simply launch on the Seq Builder icon here. And what that will do is we'll just open up that Seq Builder application. When you initially launch Seq Builder, there's that blank screen. And now what you can kind of do is you can either drag and drop data into this window, or you can simply open data. There's a number of different ways in which you can enter data into a project. So under the File pull-down, if you have a link to a specific Entree or GI ID, you can open that via Entree. You can also do a File New DNA, so File New DNA. Or in this specific case, what we're going to do is we're going to actually open up a protein and a nucleotide equivalent here pretty readily. So to do that, simply File, Open, and then I'm going to navigate to my folder in which my protein file is located. And then I'll do the same with the nucleotide. As you can see here, I have two files that are open. What I'm going to first do is kind of look at this file here and just kind of describe what you're seeing here in this interface and what you can kind of do to make this less busy or more busy, as well as save some of the default layouts if you like a specific setting. So I'm just going to maximize this nucleotide string. And I'm going to drag what we call this feature window down here towards the bottom so you can see the landscape a little better. So these are what we call curtains here on the top left-hand side, and these are the various views that you can toggle between. So in this curtain, you can toggle between sequence view, linear views, circular map reviews, primer lists, mini maps, site summaries, and so forth. One other thing that you can kind of do is I can also drag a view from the left to the right here and actually change the same thing down here. So if I don't want my feature list to be displayed here, I can change that to a linear map view, and that will be updated readily here. A highlight that I make in one selection will automatically be updated in any other views that you select. So that is a pretty helpful functionality that's built in the software. One other thing to consider or keep in mind is if you don't want all these views represented or shown, you can actually make this as simple or as busy as you want to. So if you want all the features, the translations, the open reading frames, the full translations, all to be turned on all at the same time, you have that option. You can also file and save that option as the default layout template. It's really depending on your needs and what you want to display, but you do have that option with the software. What that will do is anytime you open the software in the future, that will be your default view, that will be your default setting. You also have the option to apply a specific layout template or reset a view based on your project. So what we're going to do here is we're going to go back to that feature list. We're going to turn off some of these open reading frames, but we'll keep this features displayed on. We're actually going to minimize this nucleotide here right now. I just wanted to talk about this target, this histone that I'm working with here, this protein. So when you're setting up your expression reactions, your projects, everything obviously starts with your targets. In this specific case, I've downloaded this histone from NCBI. I know this is my coding region. This is the translated product of that nucleotide. The first steps, if I wanted to insert this protein eventually into an expression vector, is I want to basically optimize to make sure that I'm going to try to look for, minimize the propensity of possibly rare codons for whatever expression vector that I want to insert it into. To do that within the software, we actually have a number of genetic codes that you can apply based on what organism that you're working with, as well as what expression vector or host that you're using. So to change, some of the genetic codes, if you go to the sequence pull down, you do have the option to select a specific genetic code of interest. By default, we have the standard genetic code, which in the third position is going to have the wobble. So if I back translate this, this protein here, it's going to back translate into that the standard genetic code, which is going to have that IUB ambiguity. In this specific case, since I want to express this in an E. coli host, I'm going to essentially go to the select genetic code, and I'm going to change that genetic code to be a highly expressed non-gender E. coli back translation code. The end result is when I back translate this protein into the nucleotide equivalent, I'm going to have a target that's already been optimized for expression in E. coli, so thereby minimizing translation problems, truncated proteins, weakly expressed targets, etc. Okay. One other quick mention here while I'm on the genetic code part of the software is the ability to edit your genetic code. So once you go to that portion of the software, genetic code editor, you do have the option to drag and drop or change the genetic codes, start codons, based on whatever organism or whatever paper you're reading to optimize your codon expression tables. All right. So what I'm going to simply do here is I'm going to click on this feature. So this is a histone, and I'm just going to show you how to back translate this to make sure that it's optimized for E. coli. So we're going to go back to this select genetic code. I'm going to verify that yes, the highly expressed E. coli back translation code is selected. And then now I'm going to simply go to sequence, new sequence, back translate. The end result is basically the nucleotide equivalent here. And then this target would be suitable for, is ready to be, is optimized for rare codons. That doesn't have rare codons here in the sequences that it will affect the problems, issues with rare codons. I'm actually going to close out some of these here. We're finished working with this protein, and now I'm simply just going to focus on the other target that I have open here. So this is that nucleotide again. This one's a little bit more annotated than the other one, so I want to use this one. But right now I'll kind of show you some other steps that you can kind of take a look at to make sure that this target is going to be properly inserted into the expression vector of interest.
Brian, before you move on,
Yep.
You've mentioned a few times the annotations in the sequences and the feature list. Is there a way to edit those annotations and features?
Sure, Katie. Yeah, everything that you see here can be manipulated. So if I wanted to add a specific nucleotide string here, or if I want to highlight a selection and create a new feature, you have the ability to make those edits directly within the software. So to create a new feature, I would simply just highlight a selection. I would select features, and then you have the option to file new feature with translation. There's also other tools if you're trying to join features, joining exons, and so forth. There's also abilities to join features as well directly within the software.
Great, thank you.
Okay, cool. Great. The next step here is I want to take a look at the reading frame of this target. I want to make sure that we are choosing the correct reading frame, that the frame of this target is going to be obviously in frame with the actual expression vector that we're going to utilize in the next step. To kind of take a look at or evaluate frame, that's what this little folder here is called, the open reading frame. So if I expand on this folder, you have the ability to show the top strand and the bottom strand frame of this target. So the one frame is going to basically the frame that doesn't cause the target that's highlighted to shift. The two, what we know it as green here, would be the +1 shift, and the purple would be the +2 shift. So if it causes it to shift 2 base pairs, obviously that's going to affect your translation when you eventually try to express this target of interest. In this specific case, just looking at this, I see that what's listed as number 1, there's my initiating methionine. It's being translated all the way down to a terminator, or a translational stop. So I'm simply going to turn off the bottom strand and then turn off this +1 and the +2 frames here, and there's my target of interest. So as part of this, I've already had it annotated, and I know that here's my feature, there's where it starts, and further downstream that's where it stops on the 3' end. So that correlates and matches up to the open reading frame selections that we have specified here within the software. One other thing I'd like to highlight while we're talking about open reading frames is there are some open reading frame detection options built in the software. So if I go to sequence ORF detection options, if you want to specify a minimum ORF size, you have the option here, as well as if you want to show only ORFs with a start codon or without a start codon, or with or both, which we have selected here by default within the software. So what we've done here with this target is, one, we've looked at, evaluated whether there's any rare codons within this target that will affect my downstream expression of this target. So we've optimized for codon usage. Another thing is, now that we've looked at open reading frame. So we've looked at the open reading frame and made sure that what we have listed as a feature here is actually in frame of that initiating methionine. So the next step that you can kind of do, in this specific case, it's probably not as important, but if you're working with novel gene identification and you've identified maybe an open reading a frame of interest, there's also the ability directly within the software to blast any selection. So in this specific case, if I wanted to highlight that selection and if I wanted to blast that selection, directly within the software, there's the ability to blast a number of different, databases directly linked to the software. So whether it's the non-redundant, or if you want to blast a genomic ref-seq or RNA, you also have that option to verify what you're seeing, yes, is correct when you eventually insert this into your expression vector of interest. Okay, what we're going to do here now is we're going to minimize this target, and then we'll kind of talk about the expression vector that we want to look at. So to open that expression vector, I'm simply going to go to File, Open. And then in this specific case, it's a PET expression vector, so PET28B. I'm going to double-click on that, and that's going to open automatically within the software. One thing that I briefly kind of like to mention is, when you download files from various vendors and so forth, every once in a while you'll get them in the wrong orientation. So if you would go to the vendor for this vector, for example, it would actually be in the opposite orientation. So I've previously made sure that the F1 origin of replication is shown from 5 to 3'. So, and then to basically reverse that orientation, you would simply go to Sequence and then reverse complement if whatever selection that you've highlighted there. So to clarify, if you try to pull down the same vector and do these same steps, you'll need to reverse complement this. But for my purposes and my likes, I'd like to see everything in the 5' to 3', my T7 promoter at the 5' end, and my T7 terminators at the 3' end of whatever I'm looking at. What I'm going to do now is kind of just quickly navigate to the multiple cloning site of this vector, expression vector. So it's a T7 vector. It's roughly 5,300 base pairs in length. To quickly navigate it, I can scroll down the feature list here, or I can simply just expand the features, expand the alphabetical list, and simply right-click on the multiple cloning site, select feature, and then as you can see there's my multiple cloning site of this specific vector of interest. This vector has a kanamycin resistant, has a T7 promoter, LAC operator, has some histidine tags, thrombin cut sites, T7 affinity tags, and some histidine tags at the 3' end. So the next logical step, when looking at this vector, is just making sure that, okay, one, that you choose some enzymes that can be readily easily added to your target, and then also readily easily inserted into this expression vector of interest. You could take a look at the open reading frame here if you wanted to turn on that. So I'm going to deselect this feature displayed. So you can turn on that open reading frame to verify that whatever the vendor is supplying you is correct. Based on my experience with these vendors, anytime that they provide something or specify something as the multiple cloning site, they try to make it make sure that everything is correct, everything is in frame with whatever they're identifying in the multiple cloning site of their vector of interest. One thing that you can help make your job a little bit easier as researchers in picking those restriction enzymes is actually looking at some of those restriction enzymes that the vendor does recommend in their vectors. So what I've previously done here is created what's called an enzyme selector with the software. So one of the folders that you'll notice here is the enzymes displayed folder here on the left-hand side. By default, nothing is displayed, but I've turned this selector on, so that's why we're seeing it here in a previous project. But the selector that I previously displayed was what's called the PET28B selector that we've created. What this selector is just those enzymes that are recommended and present that are unique cutters in the PET28B multiple cloning site. So this will parse out all those enzymes that you have no interest in using whatsoever for this for this cloning reaction. To create those selectors, you would simply go to the enzyme pull down. I know there's a whole bunch of different options here. So the new enzyme, the enzyme manager, this would be the location to go to if you needed to add a new enzyme to the enzyme catalog that's built into the software. The enzyme catalog is directly from New England Biolab's Rebase catalog. To add a new selector, I would simply go to Enzyme Selector Manager, and that's going to open up my Enzyme Selector Manager window here. As you can see here, I have a number of other selectors that have been previously created with the software and provided with the software. So there's selectors for absent sites, unique sites, 3' overhangs, 5' overhangs, so forth, as well as customized selectors that I've created based on previous projects. To create a new selector, you would simply select New. So for example, if we are going to create PET 28, or let's just do 29 here. You would simply identify that vector or the nomenclature that you want to utilize, and then you would simply there's options to pick whether by class or overhang frequency, pick or combining different selectors. So you have that option here to specify a specific class, site complexity, giving a range, only precise cutters, random cutters, and so forth. You also have the option to choose overhang type, specify an overhang length, 5' overhang, 3', as well as specify a range. A frequency, so if you want to look at a cut frequency between 1 and 4 or 5, whatever your number would like to be, you have the option to do that. And then the one that I did previously to create that PET28B enzyme selector is I simply just picked those enzymes that were present in the multiple cloning site of PET28B based on the documentation supplied by the vendor. So you would simply add those enzymes that you would want to that selector, and then once you're finished, you would simply select OK to save that selector for future use. This is a very helpful resource if you have a vendor, a vendor that you primarily use on a regular basis for expression enzymes, and also restriction enzymes that you use on a regular basis that are in your freezer. Okay. So now that we've looked at this target, we've identified, we know that this target is in frame. We've created a selector that we know of those enzymes that are unique specifically in the multiple cloning site. Now the next step is try to take that target that we looked at previously and insert it into this expression vector of interest. So I'm going to minimize this vector. Yep.
While you're transitioning there, we have a couple questions that have come in.
Sure.
One is about predicting ORTs. Can you predict open reading frames for Sanger sequencing data in SeqBuilder, or how would you do that?
Sure, definitely. One thing I'd like to clarify here is this is just normal sequence data. So this is from a FASTA file, this is from a GenBake file that was pulled down from a Entree. You would basically be able to import whatever sequence and protein data that you'd want to in SeqBuilder as long as it is supported. So to go, you simply file open if I go to the all known formats option here, it does give you the option to bring in, GenBank, Lasergene files, EMBL files, trace data files, AB1, ABI, SCF, so forth. So yeah, you would have the ability to. What it's going to do is simply just going to bring in the nucleotide string of that Sanger data. What I would recommend doing before SeqBuilder is actually doing that config assembly in a different application that we have called SeqMan Pro. So what SeqMan Pro is going to do is going to take that trace data, it's going to assemble it into contigs, or it will basically trim off the poor quality data of if you have one ABI read, and then it's going to make sure that whatever you're analyzing within SeqBuilder, it's going to make sure that it's not poor quality reads at the 5' or the 3' end of that Sanger data. So yes, you can enter whatever data you would like to into SeqBuilder. But
Great.
if you want to use specifically the electropherograms, I would point you towards SeqMan Pro. Yep.
Thanks. And then just one more question before you move on. This one kind of going back to something you showed at the beginning. Is it possible to optimize a sequence for expression in other organisms besides E. coli? Specifically, somebody asked about mycobacteria.
Sure, yes. In that genetic code that I previously mentioned, so there are a number of other genetic codes. The one specific for you said mycobacteria?
Yes.
I don't know if there's a specific genetic code for that one built into the software, but if you have a paper that describes the codon usage chart of mycobacteria or whatever organism that you're working with, you have the option to basically change or create your own genetic codes based on information that you find out as part of your research. Yep. And you can apply that genetic code for those back translations.
Great, thank you.
Okay. Okay, so the next step is we're going to minimize this vector here. And now we're going to go back to that target that we initially were looking at. So to reemphasize, we know this is in frame. We know that's optimized for expression in E. coli. So the next step is now to basically add those appropriate, restriction enzymes to the ends of this target to be, correctly inserted into that pet vector of interest. Before we do that, let's just go back to that vector. I'm sorry for the confusion here, but we need to pick those enzymes. So previously, actually the two enzymes that I want to pick here is actually the NDE1 and then the XHO1 restriction enzyme sites. So the result of this is basically I'm going to have a thrombin cleavage site that I can use this histidine tag here for purification, and then depending on this protein, I may want to try to cut it off the column using thrombin or so forth, to where the end result is potentially almost a near native target for downstream analysis. I could also potentially manipulate the sequence string here to maybe put a terminator here before this histidine tag, and then that would be even better. But for today, we're simply going to utilize the NDE1, the XHO1 restriction sites for this cloning strategy. Okay. So we're going to maximize that nucleotide here again. And then simply I'm just going to walk you through the steps to add those inserts onto the end of that target. So I've highlighted this selection. So to highlight a selection, I can simply drag and highlight a random selection, or if I simply click on a feature, since this sequence is already annotated, I would simply click on that feature. And then now I'm going to direct you towards one of the pull downs called the priming pull down. So if I simply just wanted to highlight a selection and to very quickly create primers from a selection, that's what the second option would allow me to do. So I can highlight a selection and select top strand, and it's going to very quickly create a pretty random, a primer based on that selection. I can also do the same thing with the top, the bottom strand. What I'm going to do here is I'm going to go to priming, create primer pairs, because I want to primers combinations that are specific for this target of interest. Once I do that, the Create Primer Pairs window pops up. There's a number of different options. I'll expand these to show you them. So I can create primers that end exactly at a selection. I can choose an optimal primer range. It kind of shows me what's going to be amplified. It gives me the salt concentration. It gives me some basic primer characteristics, the primer length, the TM, unique uniqueness at 3' ends. These are all things that I can change depending on what the type of PCR application that you're doing, what your specific what organism you're working with, and so forth. For this overview, I'm simply just going to leave the default settings here because I want to create primers that will end at exactly at that target that I've selected here in the project. Okay. Once you do so, you can clearly see that there's a top strand primer that's created. If I click on this palette tool showing the little red bottom strand primer, it's going to quickly navigate to that 3' primer. I'll click back to the green one showing the top strand primer. There's also some primer binding site information that's kind of shown here. So this will show you whether there's any possible false binding sites across the target that we've highlighted, the target that's in the project, as well as where they exist, if they do exist. In this specific case, it's only showing a top strand and a bottom strand. It's going to show you the stable dimer information. It's going to show you the most stable most stable pair dimer information, as well as the hairpin information. One thing that you'll note here is based on the parameters the default parameters that we selected, these two primer combinations may not be optimal for most stable dimer formation. So what the software will kind of do is identify and tell you that this combination is actually pretty bad. They wouldn't recommend that for most stable pair dimer creation here. So to kind of alleviate that or kind of fix that, one thing that you may want to look at within the software is another tool that's up here called the Introduce a Codon Change Mutation. So there's this ATC palette tool. If I click on that, what the software will basically allow me to do is kind of look at, and try to incorporate maybe a silent mutation, changing from an AAA to an AAG, that will kind of minimize, some of the problems that you may see in regards to dimer formation and so forth. So when I do that, when I change that from an AAA to an AAG, as you can see here, that update will give me a solution that will be a little bit better for my expression. One thing I kind of like to note here is the original nucleotide string here is not changed at all. What we're doing here is we're actually just creating primers, and those primers are being saved to your primer list under this view here directly. If we go back to that primer design view on the top strand primer. So basically, the primers that we've created here, they would be suitable for, say, colony PCR. If you wanted to look at a clone and see if there's a clone that has your target of interest, these may be suitable for that type of experiment. The next step is now we want to add those NDE1 and XHO1 restriction sites at the end of our target. That leads us to this next palette tool here called Introduce Restriction Sites. So if I click on that, in previously working with this target, I know that if I cut here, it's going to give me an option for silent substitutions that I have the ability to add an NDE1 restriction site pretty readily without changing the actual target at all. So it keeps that ATG initiating methionine at the 5' end of my target. As you can see here at the top of the screen, all the different enzymes that are present are automatically populated to the screen. If you want to kind of minimize the busyness of that, you do have the option here to, if I expand on this enzymes displayed, to turn all those enzymes off. And then we can go back to that selector we created under the filter by selector option and specify PET28B. As you can see here, there's my NDE1 restriction enzyme that has been added to the 5' end of that target. Now we can actually go to the 3' end of this target and do the same thing and then adding that XHO1 site. So to do that, I'm simply going to click on that scissors. Sorry. I'm going to add the cut site right at the end. I'm going to select any enzyme, and I'm simply going to hit X, and that's going to scroll down all the way to XHO1. And then as you can see here, there's my XHO1 site that has been added to the end of this target. The next step, so we've added those appropriate sites at the end of our target. Now we've created the primers that are appropriate for adding those sites to the end of the target. One thing you can kind of do to get those primers out of this project is actually go to the primer list and then actually file if you wanted to save these as a primer catalog. export as a FASTA file, a text file, or a primer catalog Excel for future downstream applications, you can do so. Let's go back to that primer design view, click on that top strand, and then now what we're going to do right now is use the in silico capabilities of the software to amplify this target to create a clone that's suitable for cloning into my expression vector of interest. So to amplify this target, all I'm simply going to do is I'm going to click on the 5' enzyme that we've created. I'm going to navigate to the 3' end. I'm going to hit Shift, hold down Shift, and hit the XHO1. As you can see, it's going to highlight the region between those two cut sites. It's going to show me my TM information, the length, and so forth. But the next step now is to go to priming and select Create Insert by PCR. What the software has done now is it amplified the end of that target to make sure that those appropriate ends have been in silico amplified. To verify that, I can simply double-click on this amplified insert. So this is what we call a cloning project. So you also can create your own cloning projects by selecting File, New Cloning Project. You can also add your own vectors to this. You also do have some customers that like to keep clones in this type of format. One thing with this in a cloning project, you do have the ability to search through a cloning project. So if you have hundreds of clones or hundreds of vectors or hundreds of amplified inserts here, you do have the ability to edit, find, and you can actually do some searches for sequence names. You look for a specific nucleotide string, feature name, and so forth directly within the application. So I'm going to simply double-click on this amplified insert. We're going to go back to that enzyme displays folder. We're going to expand on that filter by selectors, and we're going to show you that PET28B multiple cloning site selector that we've created. I'm going to drag this feature window down just to make it a little bit more visually appealing here. As you can see, there's my NDE1 site that has been added to the 5' end of my target. There's my feature. There's my initiated methionine. I can confirm this target even further by turning the ORFs on. I can blast this to make sure everything is in frame. And there's my XH1 site there at the 3' end. The next step is I'm simply going to now perform the traditional cloning interface to get this into my expression vector of interest. So I would simply double-click on the 5' end of the restriction enzyme and shift-click on the 3' XH01 site. And then I would go to the cloning copy restriction and selection. What's that going to do is it's going to put it in memory, and now we can go to our expression vector. We're going to go to that multiple cloning site again. We're going to select those same two restriction enzyme sites. I'm going to double-click on the ND1, shift, double-click on the XH01, hold down shift, and double-click on the XH01 site. Then I would go to cloning, clone restriction inserts. What pops up is a clone insert dialog box. What this is kind of showing you, it's showing you the left end of the 3' end of that vector, as well as the 5' end of the insert. It's also showing you the right end, the 3' end of the insert, and how that matches up with the 5' end of the vector. If everything matches perfectly and you have a appropriate cloning strategy, you'll see checks here. If for some reason, if the strategy that you chose, say you didn't you didn't select the restriction enzymes correctly, there will actually be a check here, and it'll say, oh, it's unsuccessful, it's not matching. The software does give you the option if you want to automatically fill in or trim, or if I simply want to drag some of these arrows, 5' and 3' to force cut it, you can do so here. To complete this cloning insert, I would simply select clone, and then actually a new clone is created that has my target that has been inserted into my expression vector of interest. So to visualize some of that, I can go to the linear map and it will show you, there's my parental information for my expression vector. There's my information and annotations for my target. I can go to the circular map to view those as well. Okay.
Brian, we do have a couple of questions that have come in. Sure. The first is about vectors. Is it possible to import your own vector data into SeqBuilder to use for cloning experiments?
Sure. Yeah. Built into the software, actually, we actually have a vector cloning catalog. So with any installation of Lasergene, we actually have training data as well as vector catalogs that are automatically built into the software. So as you work through some of the resources that we provide online as well as within the software, it will point you towards demo data that you can kind of utilize to get a better understanding of what the software can specifically do. So for example, there's a cloning vector option here. So in the next in silico cloning interface, we'll go through some of the TA cloning and kind of show how those TA cloning vectors are pulled from the cloning catalog. But yeah, you can insert whatever you would like into this catalog for further downstream use.
Great, thank you. And then a question about primers. It looked like when you were creating the primers for your target sequence, you used the default options, and that created one set of primers. Is that the best pair, and is there a way to create more pairs, or can you just explain those options a little bit?
Oh, sure. With the software, I'll expand that initial target here and kind of do the same project again. So there's a number of different parameters you can kind of change. You can also, it does give you the, if you want to choose an optimal, it does give you the option here on the left-hand side to show alternates, specify alternate primers. So you can change any of these default settings as you see fit. So if you want to stay within a certain base pairs of selection, display at most, in this specific case, when I select the primer stand exactly at that position, it's only going to give me one primer combination to choose. The primers that are created are based on the nearest neighbor method. So the these are the I would say based on that method, these are the primers that they would recommend. If you would want more options, what you would probably want to do is specify an optimal primer location. So stay within not a certain number of base pairs of selection, base pairs of whatever selection you've identified, as well as it does give you the option to see alternate pairs. So if you want to specify 10 additional primer pairs, you do have the option. You would simply select choose optimal primer location. You could say it's within 0 base pairs, and it does give you some options to see other primer combinations.
Great, thank you.
Okay. All right, we're going to maximize this window again here. All right, we're going to turn off close some of these windows just to minimize the busyness here. The next thing I thought would be kind of helpful to go through is a simple a TA cloning application. So we're gonna start with that initial sequence that we worked with here. Same sequence, same target, but we're simply going to highlight it and kind of show the abilities to use the in silico cloning wizards for for TA cloning. There's also cloning wizards for gateway cloning, gateway multi-site cloning, directional topo. But for today's presentation, I thought it would be helpful just to briefly go over the TA cloning, since the other workflows are pretty much similar. We do also have a number of videos online. So gateway cloning, the multi-site gateway cloning application is sometimes a little bit more intensive, but you can look at that video online just to show the logical workflow for that. So to do a gateway clone, there's directional TA clone. There's essentially 2 steps. There's the creation of that insert, so amplifying the ends of that insert so it's appropriate for TA cloning. And there's the actual inserts into a T vector of interest. So what I would simply already done here is I've highlighted that selection that I want to insert into a TA vector. I'm going to select Create TA Cloning Insert. And then what's going to come up is our TA Cloning Advisor. So that cloning project is created again. There's my amplified insert. And then basically what the software is going to giving you the option to do here is it's giving the option to choose a number of different linearized T vectors that are already built in the software. You also have the option to add other vectors that are not included in this list by adding them to that cloning vector catalog that I showed you there previously. So if we simply pick the first one, PBAD dio topo, hit clone, what's created here is essentially the forward and the reverse clones orientation of that with that target that has been inserted. So this is correct with TA cloning. You can't control the orientation, so you're going to see a forward and reverse orientation. Whereas in directional topo gateway, obviously you can establish and make sure the orientation is correct. So to look at that vector here, you simply double click on it again. I'm going to drag that feature list down, and as you can see, the appropriate primers have been created that will add those ends on, those appropriate ends to be compatible with TA cloning. A couple other things while we're just simply looking at this interface is, you also have the ability to change, some of your translation displays. So, if I wanted to turn translations on and see how all the translations appear, in these various clones, these are things that, are present in typically, any application that you take a look at. You can change, the translation display under the view, the format option. Say if you wanted to change your residue grouping, and this may assist with maybe seeing if things are in open, in reading in frame with whatever approach that you're looking at. You can group by 3 as well as group by 10. So these are all things that are somewhat helpful for evaluating whether whatever option you choose for cloning is in frame with your target. Okay. So the other logical steps, with the software, is you also have the ability to do your directional topo and your gateway sites. One thing I would recommend is if you're interested in those applications, I would direct you towards our videos because those are a little bit more informative and a little bit more thorough in regards to overview of those different applications. Okay, that pretty much concludes the primer design and cloning for today. Was there any questions out there?
Yeah, we do have a couple questions, and now is a great time to chat in any more questions. So if you do have questions, chat them in now, and we'll try to get to as many as we can. One question that came up early on, and I thought maybe you could use this time to sort of explain this, the cloning capabilities in Lasergene, is that included in Lasergene Core Suite, or are there other tools that are needed to do this?
Sure. Let's just go back to the navigator, and I'll point you towards some of the different solutions that we have here. So Lasergene is actually a number of different tools. Roughly there's 10 different tools or functionalities that people should be aware of. So we have a number of different packages. Our base package is what we call the cloning configuration within Lasergene. That configuration would include Seq Builder, which we went through today. Primer Select, which is another application all dedicated to primer design. There's some additional functionality in Primer Sign for mispriming. There's some alternate views and reports that you can generate. I would point you towards that if you want a little bit more options in regards to mispriming as well as report generation. And then the other application would be our EditSeq application. EditSeq is more of a legacy program for our long-term, for our customers who've been with us, since our inception as a company, essentially. EditSeq is a very simple nucleotide viewer, but for all intents and purposes, all the functionality exists in Seq Builder. Yep. There's also other applications. There's the alignment suite. So SeqMan Pro Megaline would be added to that base configuration that I mentioned to basically turn into the alignment suite. And then we also have what's called the full Lasergene suite, which has the Protean, Protean 3D, GeneQuest, GenVision, SeqNinja applications. We do also have other applications outside of Lasergene. Those applications would be, so for example, SeqMan Engine. That would be our tool specifically for next-gen sequencing. So if you're working with Ion Torrent, Illumina, Roche 454, this would be the assembler that will assemble that data. If you're working with those platforms, I would recommend looking at Engine. ArrayStar at its core is a tool for microarray analysis. So if you're working with AFI, Illumina, those microarray platforms, NimbleGen. And then there's also components that will allow you to do your RNA-seq, ChIP-seq, microRNA experiments. So if you want to normalize that next-gen sequencing data, quantitate a probe set of interest or a genome of interest, it does give you some tools to do some of those next-gen workflows. Another application in here is MAUVE. MAUV is another tool that we can provide. It's actually a tool created by one of Dr. Blattner's colleagues. It's a genome aligner. So if you want to align 4 or 5 E. coli genomes head-to-head and you want to view the differences or the translocations or the similarities across those genomes, it is a pretty unique tool that allows you to do that.
Great. And then a question about the restriction sites. Somebody asked, is it true that I have to select restriction sites within the multiple cloning site of the vector to do cloning in SeqBuilder?
No, no. You have the option to select whatever restriction sites that you would like to, select. But based on my experience, if you're working with an expression vector from a specific vendor, they've already specifically optimized and made sure that whatever restriction enzyme that they've selected, is in frame of that promoter that they're recommending for expression. But you have the option to, I can select whatever enzymes that I want to apply to my project to be used for cloning. What's very important, whatever enzymes that you select, is one, you want to make sure that's in frame with whatever promoter that you're using for expression, and then two, I would really make sure that you look at the codon usage charts to make sure that that's optimized for whatever expression strategy that you're utilizing. But yeah, you have the ability to select whatever enzymes that you want to display as well as use for whatever cloning strategies that you want.
Great. And then another question, is it possible to create an antisense construct using the software?
Okay. So right now, this is if you wanted to create an antisense, I can simply highlight a selection and do a reverse complement. In the cloning wizard, I think there's also a palette tool that gives you the option to focus on to automatically create the antisense as well. So in that, as part of that cloning wizard that popped up that looked at the 5' ends of your vector as well as your 3' as well as your insert, it does give the option, it gives you some options to do a reverse complement as well there.
Great, thanks. And then last question, kind of going back to the other applications in the core suite that you were talking about, if you wanted to align your target sequence with the second sequence, what application would you use for that?
Sure. I would actually use our SeqMan Pro application here. So I can very quickly go through that logical application, that approach. We have this is demo data that's built in the software that we'll be utilizing. When you initially open SeqMan, there's 2 windows. This is called the unassembled sequences window. This is where your contigs that will be aligned to. So you simply go to add sequences. Here's some demo data I previously worked with that's built in the software. This is SNP demo, but for this specific case, it's similar to the experiment that you're trying to do. What I would do is I would simply mark that first, whatever you're listing as the wild type, or if that's the initial plasmid and you want to assemble the second, the sequencing reaction against that plasmid, I would mark that as a ref. If I double-click on the sample, briefly, there was a question about bringing data into SeqBuilder, specifically from a big dye Sanger reaction. What it's going to do is, when you bring it into SeqBuilder, it's simply going to bring in the nucleotide string. If you wanted to view these electropherograms, that's why we've developed SeqMan Pro. That gives you the option to view those electropherograms. It actually also will look at the quality of the individual peaks and assign quality scores built into the software. So to compare that sequencing reaction against that wild type, I would simply mark the one as the reference, hit assemble, and basically there's my contig that has been generated. There's my reference, there's my sample. So if the reference is annotated, there's my annotated reference, and then I can expand on this little triangle in the alignment view to view my trace files. So right now, this is what we call the alignment view. This is one of the views you can kind of take a look at, that alignment. You can also go to what we call the contig strategy view, and obviously we'll show you any differences. I can show any specific conflicts that exist across that sequence. So if I zoom in, actually get to the individual conflicts. Or one other helpful tool is actually from our SNP pull down. So whether you're actually specifically working with single nucleotide polymorphisms or just looking at ambiguities, if I go to the SNP report, it will show you those ambiguities or differences that exist between the reference and the sequence. So if I expand on this, what this SNP report is kind of showing me, it's showing me the actual, contact position. So if you're saying a reaction maybe went over the ends of your reference, it's going to actually show you the contact position. It's also going to show you the reference position. So if you know at position 10 in this specific case of that wild type that you're looking at, there should be or should not be, an ambiguity or a difference or a SNP. This will kind of show you that in the report. I can also double-click on any of those entities, and it'll directly lead me to that alignment view where I can confirm this. Maybe it's potentially a sequencing artifact, or maybe it is a true SNP that you may want to further evaluate further. But there's also codon usage information, how far it's from the codon distances. And then this tool is also helpful for if you're working with next-gen sequencing. This is the end report of our Segment Engine application where you can look at Bayesian SNP calculations and so forth. So I'm going to actually close out of Segment Pro. I think that pretty much answers the question. But I would point you towards Segment Pro for that.
Great, thank you. With that, that's actually a great segue into our next webinar on August 22nd, which is 3 weeks from today. Matt Kaiser will be talking about assembly and analysis tools for next-gen sequencing data, specifically de novo genome assembly. And then after that, I don't have it up on my slide here, but we'll have another webinar on September 5th, which is another Lasergene Core Suite webinar where we'll talk about sequence alignment and gene discovery tools in Lasergene Core Suite. So 2 very interesting topics, and hopefully we can expect to see some of you at those webinars as well. To sign up, just go to our webinars page, dnastar.com/webinars, or you can contact me at webinars@dnastar.com for more information. And I also want to mention something that Brian touched on a little bit in his presentation was that we do have lots of videos on our website. We have over 100 videos, and we come out with a new one every week. And these are short videos, 2 to 4 minutes usually, that show a specific workflow. So a lot of the questions that we had today, we may have videos on those workflows with an in-depth step-by-step instructions on how to accomplish those objectives. So that address is dnastar.com/videos, or you can look up DNASTAR on YouTube and get all of our videos that way as well. So thanks again to all of you for coming today. And if we didn't get to any of your questions, we'll follow up with you after the webinar. And again, if you come up with other questions later, just send us an email and we'll get back to you soon. That concludes our webinar today. This webinar will be posted on our webinars page, so if you missed any part of it, you can go back and watch it again.